Ionic properties of amino acids and proteins. 20 amino acids varying in size, shape, charge, and chemical reactivity are found in proteins, and each has at least one codon in the genetic code. 19 of the amino acids are alpha amino acids. The amino acid and carboxyl groups are attached to the carbon atom that is adjacent to the carboxyl group with the general formula RCHNH2COOH, where R is an aliphatic, aromatic, or heterocyclic group. RCH bracket NH2COOH. Okay. Where R is an aliphatic, aromatic, or heterocyclic group. The only exception to this general formula is proline, which is an amino acid in which the NH2 group is incorporated into a 5-membered ring. With the exception of the simplest amino acid glycine, R is equal to H. All the amino acids found in proteins contain one asymmetric carbon atom and hence are optically active and have been found to have the L configuration. For convenience, each amino acid found in proteins is designated by either a 3-letter abbreviation, generally based on the first 3 letters of their name, or a 1-letter symbol, some of which are the first letter of the name. Since they possess both an amino group and a carboxyl group, amino acids are ionized at all pH values. They are ionized at all pH values. Neutral species represented by the general formula does not exist in solution irrespective of the pH. When increase pH, net negative charge will increase. Increasing pH, net negative charge will increase. And when net positive charge, pH will be decreasing. Net positive charge COOH gives a net positive charge in the lower pH. Then COO minus 0 net charge. That is a switcher ion case. COOH gives lowering of pH, net positive charge. COO minus gives net negative charge. That means increase in pH. Thus, at low pH, values on amino acid exist as a cation, and at high pH, values as an anion. At a particular intermediate pH, the amino acid carries no net charge, although it is still ionized, and it is called a switcher ion. It has been shown that in the crystalline state and in solution in water, amino acid exists predominantly as this switcher ionic form. This confers upon them physical properties characteristic of ionic compounds. Ionic compounds. High melting point and boiling point, water solubility, and low solubility in organic solvents such as ether and chloroform. The pH at which the switcher ion predominates in aqueous solution is referred to as the isoionic point, because it is the pH at which the number of negative charges on the molecule produced by ionization of the carboxyl group is equal to the number of positive charges acquired by proton acceptance by amino group. In the case of amino acids, this is equal to the isoelectric point Pi, since the molecule carries no net charge and is therefore electrophoretically immobile. In the case of amino acids, this is equal to the isoelectric point, since the molecule carries no net charge and is therefore electrophoretically immobile. The numerical value of this pH for a given amino acid is related to its acid strength pKa value by equation Pi is equal to pKa1 plus pKa2 by 2. Pi is equal to pKa1 plus pKa2 by 2. That is the relationship between Pi, or the isoelectric point, and acid strength. where pKa1 and pKa2 are equal to the negative logarithm of the acid dissociation constant Ka1 and Ka2. In the case of glycine, pKa1 and pKa2 are 2, 3 and 9.6 respectively, so that the isoelectric or isoionic point is 6.0. At pH values below this, the cation and zither ion will coexist in equilibrium in a ratio determined by the Henderson-Hasselbalch equation, whereas at higher pH values, the zither ion and anion will coexist in equilibrium. For acidic amino acids such as aspartic acid, the ionization pattern is different owing to the presence of a second carboxyl group. So cation will be there, zither ion form, and then anion, then the anion, two net negative charges. In this case, the zither ion will predominate in aqueous solution at a pH determined by pKa1 and pKa2, and the isoelectric point is the mean of pKa1 and pKa2. In the case of lysine, which is a basic amino acid, the ionization pattern is different again, and its isoionic point is the mean of pKa1, pKa2 and pKa3. As an alternative to possessing a second amino or carboxyl group, an amino acid chain may contain in the R of the general formula a quite different chemical group that is also capable of ionizing at a characteristic pH. Such groups include a phenolic group, guanidino group, imidazolyl group, histidine, and sulfhydryl group, which are the phenolic group, guanidino group, imidazolyl group, and sulfhydryl group. It is clear that the state of ionization of the main groups of amino acids, acidic, basic, neutral, will be grossly different at a particular pH. Moreover, even within a given group, there will be minor differences due to the precise nature of the R group. These differences are exploited in the electrophoretic and ion exchange chromatographic separation of mixtures of amino acids, such as those present in a protein hydrolysate. Proteins are formed by the condensation of alpha amino group of one amino acid within the alpha carboxyl of the adjacent amino acid, with the exception of the two terminal amino acids. Therefore, the alpha amino and carboxyl groups are involved in peptide bonds and no longer ionizable in the protein. Amino, carboxyl, imidazolyl, guanidino, phenolic, and sulfhydryl groups in the side chains are, however, free to ionize, and of course, there will be many of these. Proteins fold in such a manner that the majority of these ionizable groups are on the outside of the molecule, where they can interact with the surrounding aqueous medium. Some of these groups are located within the structure and may be involved in electrostatic attractions that help to stabilize the three-dimensional structure of the protein molecule. The relative number of positive and negative groups in a protein molecule influence aspects of its physical behavior, such as solubility and electrophoretic mobility. The isoionic point of a protein and its isoelectric point, unlike that of an amino acid, is generally not identical. This is because, by definition, the isoionic point is the pH at which the protein molecule possesses an equal number of positive and negative groups formed by the association of basic groups with protons and dissociation of acidic groups, respectively. In contrast, the isoelectric point is the pH at which the protein is electrophoretically immobile. That is the pH in case of proteins, electrophoretically immobile. In order to determine electrophoretic mobility experimentally, the protein must be dissolved in a buffered medium containing anions and cations of low relative molecular mass that are capable of binding to the multi-ionizable protein. Hence, the observed balance of charges at the isoelectric point could be due to partly to being more bound mobile anions or cations than bound cations or anions at this pH. This could mask the imbalance of charges in the actual protein. In practice, protein molecules are always studied in buffered solution, so it is the isoelectric point that is important. It is the pH at which, for example, the protein has minimum solubility, since it is the point at which there is the greatest opportunity for attraction between oppositely charged groups of neighboring molecules and consequent aggregation and easy precipitation. So, as a quick summary, Amino acids are dipolar switcher ionic ampholites. In aqueous solution at a neutral pH, that is the physiological pH, amino acids are ionized and predominantly dipolar switcher ionic and amphoteric electrolytes with no net charge. They are dipole ions in having two oppositely charged poles, switcher ionic in that they can behave as anions as well as cations, and amphoteric in having the properties of both acids and proton donors and bases, proton acceptors. They will be electrically neutral since their positive and negative charges cancel each other. Apart from dipolar amino acids, neutral and transient forms also exist in aqueous solution. Amino acids will be cationic in acidic solution and anionic in alkaline solution. Thus, amino acids can carry a net zero, negative or positive charge depending upon the pH of the medium. The acidic groups of amino acids include carboxyl group COOH and protonated alpha amino group NH3+. Basic groups include dissociated carboxyl COO- and dissociated alpha amino group NH2 group. Since amino acids are ampholites with carboxyl and amino groups, they can react with acids and bases to form salts. In these reactions, their carboxyl group donates proton or their amino groups accepts proton. The amphoteric nature of amino acids is biologically important because it enables them to act as buffers in solutions to resist changes in pH. In this role, they donate H plus ions as pH increases and accept H plus ions as pH decreases. The state in which the net charge of an amino acid is zero is called isoelectric state. The pH of the isoelectric state at which amino acids are electrically neutral with no net charge is called isoelectric point, isoelectric pH or isoionic point. At isoelectric point, amino acids and proteins will be water soluble and all their constituent groups will be ionized. Still then, the amino acids carry no net charge since opposite charges cancel each other. If an amino acid is placed in an electric field at an isoelectric point, it will not move either to the anode or to the cathode. Therefore, at isoelectric point, amino acids will have no mobility and their solubility and buffering capacity will be minimal. In a dipolar amino acid at isoelectric point, the amino group is basic, protonated, associated, and cationic in case of amino acid. On the other hand, the carboxyl group is acidic, ionized, dissociated, and anionic, and it forms a carboxylate ion. In acidic solution, the amino group is ionized and the carboxyl group is unionized. In alkaline solution, the amino group is unionized and the carboxyl group is ionized.